Hello, my name is Darlene Watson, and I would like to discuss can PCR and HRM be more effective than the Sanger method when it comes to finding the BRCA1 and BRCA2 gene that is found in ovarian and breast cancer. Um, we found that the outcomes are better if we can find those mutations as soon as possible. And there are two methods that I would like to discuss as a comparison to the Sanger method, which is the golden standard for doing BRCA1 and BRCA2 mutations right now. The same, all three of these tests are pretty comparable in sensitivity and specificity. And through the media, of course, you know the BRCA1 and BRCA2 gene mutations are very helpful and very useful in the diagnosis of breast cancer and ovarian cancer. So the choices that what we can do diagnostically are there for us. Okay, the average turnaround time for genetic testing is approximately 2 weeks. The new test that I want to discuss, which would be PCR and HRM, you can get those within a couple days compared to 2 weeks. And I'll discuss some of the cost savings that you can have. But first I would like to talk about the golden standard, which is the right here, and it is the Sanger method, which is a very good method. Like I said, it is costly and takes about 2 weeks to be done. But it is very, very sensitive and very accurate. So what the Sanger sequencing method does is when DNA polymerase copies a single strand of DNA, through the process of copying, usually chemical altered bases that are A, T, C, and G in the fragment are put together in a sequence for the original. Okay, so here is a piece of DNA. So you have this reaction mixture. Okay, it goes in with a primer and a template. Goes through and it makes all these copies right here. Then it goes through another process where it goes through electrophoresis so it can separate the DNA fragments, and then it goes to a detector, and then it goes to the laser detections of fluorochromes and computational sequence analysis. So you start here, and then by the end you would have some analysis on the computer. Okay? So the second method that is being compared is the polymerase chain reaction. It analyzes a short sequence of DNA or RNA into even samples of very small amounts of DNA and RNA, and amplifies selected sections of DNA and RNA and generates thousands of copies of either one. This method is performed and completed first before the HRM method is done. Okay, there is a piece of equipment out there called the QuantStudio 6 PCR system that can have results as soon as 30 minutes, and now they have the technology to add HRM with it, which is high resolution melting. Okay, so this PCR is used widely in all labs for a variety of tests. Okay, and now, what would you do if you could scan a method that quickly determines BRCA1 or BRCA2 genes? Well, we have that method. It used to be called the standard melting curve, but it is different, and it's called high resolution melting analysis. And of course, the diagram below shows the process of PCR. So you take the original double-stranded DNA, you break that apart, then it goes through all these primers and makes copies. Thousands and thousands of copies. By the end of that, then we'll be ready for your high resolution melting. Of course, you know PCR is a big part of testing in the clinical lab. Okay, so it uses, what we want to use it with is HRM to screen for mutations in the BRCA1 and BRCA2 genes. The methodology of PCR is used for making a large number of copies and short sections of DNA from a very small amount of DNA. And it amplifies and enables specific genes to be detected, such as BRCA1 and BRCA2 genes. So this process is the start of being able to find out if there are any mutations in either of the genes. Okay, so along with PCR, we have the third final step, which is the high resolution melting, which is performed after PCR is already completed. Okay, so HRM is not the same as PCR. Its methodology has higher temperatures and specific dyes that make it completely different for the test of the mutations. Okay, so the major advantage for HRM is there is no sample that is going to be contaminated because it's a closed tube method. Okay, so it's not going to be open and tested and open and tested. And the testing performed by PCR amplifies the DNA sequence so that when testing with HRM, the DNA is saturated with a piece of special dye and additional heating is applied to the sequence for the detection of any mutations that are present. So it's got special dyes for the HRM, and it goes to a higher melting resolution to find those genes compared to what used to be just the standard melting curve analysis. So it is something different and better. Okay, so what HRM is based on is the PCR dissociation curve analysis. So HRM can analyze the difference in their composition, their length, and the strand complementarity. There are 3 different characteristics that set it apart from the standard melting curve. Okay, it uses brighter dyes at higher concentrations. Okay, it requires instruments that collect fluorescent data at finer temperature resolution, and it requires software that has fluorescent scaling algorithms and plots. So the methodology uses a region of interest that is amplified by the PCR, and in the presence of fluorescent dye that binds double-stranded DNA. After PCR is completed, the product is slowly melted, and the amplicon is measured on an instrument that generates a characteristic curve. So the melting profile will display the mix of amplicon present, such as length, sequence, and GC content. The results can provide information for mutation screening, genotyping, and methylization. 2 profiles are determined: the melting curves that are similar in shape but are different from each other by melting temperature of the amplicon. The next one is the melt curve displays a distinct curve from a homozygous melt curve. This can happen from having base pairing mismatches. So as you see right here, you have the red, which is a homozygous with guanine. Okay, you have the green here, which is homozygous with adenine, and then you have the blue, which is heterozygous. Okay, and the annealing temperatures are from 77 to 87. Okay, and so here's a piece of machinery here that will start with PCR. It does its calibrations. Okay, then here, step 2 designs your PCR primers and optimizes PCR reaction. In step 2, it performs PCR. Okay, so it's going through all of its steps with the reagent. Then once PCR is completed, then it melts the samples in real time. So then it starts doing its melting curve. And then at step 5, analyzes melt data and output profiles. So as you can see, it's a very detailed, but you can see how quick it would be, how everything is just completely closed and nothing is open. So there is no way it can be contaminated. Okay. So, and then in step 2, where you would design your reaction depending on what you're looking for. Okay, in our case, we're looking for mutations in BRCA1 and BRCA2. So new dyes available will be put on the instrument. So the dyes that are specific for BRCA1 and BRCA2 will be put on this. And then the instrument the instrument uses very small amounts of reagent. Okay, so you don't use much reagent. So that's another plus. Okay, and then there are 2 validation studies that were conducted under the guidelines of Peter MacCallum, Ethics of Human Research Committee. The first study included 266 samples that were tested by biodirectional Sanger sequencing and were screened with HRM using a 384 plate well. The operator did not have knowledge of any kind of mutations. Okay, and until HRM had been performed and analyzed, all variants, including heterozygous SNPs, were considered, with having 22,797 amplicons analyzed, which is quite a bit. Of that number, 7.5% needed additional sequencing. Okay. to identify the variant, and 92.5% were considered to be a wild type. All the 35 pathogenic mutations were identified by sequencing in HRM, which was 13% of the DNA samples. So the calculated sensitivity for the detection of the variants is 99.8%, including SNPs, and the value was 99.98%. So that was very, very good. And a second validation was performed rescreening 118 archival samples, which was previously reported negative to the germline mutations with BRCA1 and BRCA2. There were 11,092 fragments that were analyzed, and 92.7 were of the wild-type amplicons, and 7.3 required more sequencing. Sensitivity as being at 100% and specificity was 99.72%. The positive predictive value was 98.98%, and the negative predictive value was 100%. So no amplicons were falsely negative. So everyone that was positive was truly positive. Okay, so then it shows you the advantages of this right here is, as you can see, this is a normal curve for HRM resolution melting, and this is an abnormal one, which would have the BRCA1 and BRCA2 genes. Okay, so the advantages to having this would be that it takes less time, and it takes a little bit of reagents, and it is very, very cost effective. So I hope I gave a good presentation, and I do believe that PCR and HRM is the way to go today, because it is cost effective and the turnaround time is faster, so the patient can be treated faster, which a lot of times they're waiting to have some kind of procedure done, which would be either a mastectomy or some kind of radiation or chemotherapy. So if we could hit those targets specifically for BRCA1 and BRCA2 genes, then we can really help the patient with a better outcome. So I would just like, again, to say that I really believe and have proven that the PCR and HRM would be more effective than the Sanger method, only because of the cost and the turnaround time. So thank you for listening.